Let's continue with mutations. They are changes in the genetic material, permanent changes, that is, in the base sequence of the DNA. Mutations can be neutral, they can be beneficial, or they can be harmful. Mutagens are agents that cause mutations or increase the rate of mutations. Spontaneous mutations, they are naturally occurring due to errors that were made in the DNA polymerase, for example, or in the RNA polymerase. So here we're going to continue with types of mutations. Let's start with a point mutation. That means a one base substitution, so a change in one base in the DNA. An example of that is shown here. So the parental DNA shows here that the sequence should be ACA, and over here TGT, and then the normal sequencing here would be in the base pairing is TGT, and instead here there was a mistake was made, and the G didn't pair with C, but it paired with T, and that mistake for some reason was not recognized, it was not proofread, and so this T right here ended up staying. And now in the next round here of replication, then we're going to have the T that was here ends up base pairing with A, and now we have the new DNA strand reading TAT, which is transcribed into messenger RNA as UAU, and where it should be UGU. So based over here, the G should have base paired with C, so the G with the C right here, and that should have led to a messenger RNA codon reading UGU, which translates into a cysteine, but instead we're getting now the UAU, which translates into tyrosine. So now we have one amino acid change, and that can have a big effect. It may or may not, but if it's a cysteine to tyrosine, it probably will have an effect, but it's just one nucleotide change, so it's a point mutation, one base substitution, and we're substituting now the amino acid cysteine for tyrosine. The missense mutation is basically one of those base substitutions where base substitution results in a change of an amino acid, and here's another example given of that. So in panel A up here you have the normal DNA, the template strand, and transcription leading to normal mRNA and translation into this amino acid sequence of methionine, lysine, phenylalanine, glycine, and a stop codon. Now we have a missense mutation down here, and that means that the TA here, that is the mutation, and now the mRNA, the codons, they read, they translate into methionine, lysine, phenylalanine, and then serine instead of glycine. So the glycine is replaced with serine, and then we have a stop. So we have one base substitution. Worse than a one base substitution and one amino acid change would be a nonsense mutation, where basically we have a frame shift. So now the base substitution results in a nonsense stop codon, and the whole protein doesn't make any sense at all. So here would be an example of that. We have the normal template strand in panel A, so that's over here. So the transcription leads to this mRNA right here, and the translation to the amino acid sequence of methionine, lysine, phenylalanine, glycine, and stop codon. And now we have a nonsense mutation that leads to an early stop, a UAG, that is a stop codon, and really all that's left is a methionine, a single methionine. Really there's no more protein made because this is a stop codon. So that would be a nonsense mutation or a base substitution mutation that results in a nonsense stop codon. Moving on to the frameshift mutation, that's probably the worst kind of mutation because it shifts the entire translational reading frame. That means you've either lost or gained one or two nucleotides, and that shifts the reading frame of the three nucleotides, the three nucleotide codons, and makes every subsequent codon an error 1. So here is an example of this. And so you have here the normal sequence of the DNA, and then transcription leads to this mRNA right here, and translation leads to this sequence of amino acids. Now we're going to take one nucleotide out in this case. So we're going to take this adenine nucleotide out, and now instead of having the 3 AAA right here, you're going to have only 2 A's and then a C right here, and it will shift the reading frame. So instead of having AAA translate into a phenylalanine, we're going to have AAC translate into a leucine. And then the next codon will be CGA right here, which was going to be CGA up here, and it should have been a CCG that translates into a glycine right here after transcription and translation, but we ended up with this alanine right here because it was a CGA that translated here into an alanine. So that's a frameshift mutation, where after the mutation, every subsequent amino acid is going to be different. So the methionine and lysine stay the same, but then here it continues with phenylalanine, glycine stop, and here we have leucine, alanine, and who knows what's going to continue. Now, chemical mutagens, those are substances, chemicals that increase the rate of mutation. There's a naturally occurring rate of mutation, but it's very small because DNA polymerase has a great proofreading function. It's very high fidelity in the enzyme. But then there are chemicals out there that increase that rate of mutation. Let's start with a few of them here, right? Nitrous acid causes adenine to bind with cytosine instead of thymine. Nucleoside analogs, those are also chemical mutagens. They incorporate into the DNA in place of a normal base, and then they cause mistakes in the base pairing. And here is an example for that. Oxidation of nucleotides making a mutagen. We're oxidizing here the adenosine nucleotide, and it normally should base pair with thymine, or if it's messenger RNA, it would be a uracil nucleotide, but then this altered adenine nucleotide will hydrogen bond with a cytosine nucleotide, which it shouldn't do that. Here's an example of how oxidation can cause mutations. And so here we're using nitrite to oxidize this adenine nucleotide here. So now it's an oxidized adenine. And now instead of base pairing with thymine, it will base pair with cytosine. And so now we have a change in the DNA, which is a mutation, and it will be propagated through cell divisions. And so now we're going to have an AC bond right here. And that leads to, most likely, to an altered amino acid sequence, and then to an altered protein with altered function. On a side note, you've probably heard about antioxidants and how they're supposed to be good for you. Well, here's the reason why. Because antioxidants, they prevent oxidation of DNA, for example, and so if you prevent the oxidation of nucleotides, then you could prevent some of these mutations, and that could protect you potentially from things like cancer and some other problems that are related to genetics. Here is some examples of how nucleoside analogs can actually cause flawed base pairing. So here we have 2 aminopurine that's incorporated into the DNA and in place of an adenine, but it can pair with cytosine. So an AT pair becomes a CG pair, and of course that's a mutation. Here's another example of a 5-bromouracil that's actually used as an anti-cancer drug because it's mistaken for thymine, and then it causes the AT pair to become a GC pair. Now you might wonder why that would be an anti-cancer drug. Well, cancer cells divide very rapidly, and so if you basically try to kill the cells by mutation, that would be the way to go. So you would mostly affect fast dividing cells. Now, this is not to say that this doesn't have any side effects. It will have for any fast dividing cell. But here's an example given of how a drug can be used to slow down the cell division of widely reproducing cells. Now, the next thing here would be radiation. Radiation is known to cause changes to the DNA, and especially ionizing radiation. So that would be X-rays, gamma rays. They basically oxidize nucleotides, and they can break the backbone of the DNA, basically. And also UV radiation, as in intense sunlight, can cause thymine dimers. And so let's take a look at this. So radiation, well, cells have typically some mechanisms to repair radiation damage, especially when it comes to UV radiation. So photolyases, those are enzymes that can separate thymine dimers that were caused by too much UV radiation. And then nucleotide excision repair enzymes, they can cut out damaged pieces of DNA and replace it with new ones that are undamaged. So here would be the typical example of a thymine dimer. So here you are going out there to the beach, if the beach is open, and you're exposing yourself to massive amounts of UV radiation. It will hit your skin, and it will cause inevitably thymine dimers. So these two thymines right here, they're next to each other, and they start to have a covalent cross-linking that's due to the UV radiation, and that's called a thymine dimer. So now they're bulging up, and now the DNA polymerase would have a roadblock ahead of it, and it wouldn't be able to get across this, so it would cause problems. And how can you fix it? Well, there's a couple of ways you can fix it. Probably the most intuitive way would be to cut out the damaged area, and that's shown here. So you're just going to cut here and here, cut out the damaged area and replace it with new DNA, and there you go. And then you don't have to deal with this problem. We all have these DNA repair mechanisms. There are some people that don't have them. There's a disease, a genetic disease that's called xeroderma pigmentosum. And these people, they lack this mechanism of fix these thymine dimers that are due to UV radiation. Those are people, they will have to live in the dark for the rest of their lives, for their entire lives. If they're even exposed to moderate, just regular sunlight, not even just going out there in full sun, but they will have on the order of like 50 to 100 skin cancers per year. So these repair mechanisms that we have, they do work, and if they don't work, then you do have the consequences. But good advice to always take some sunscreen when you're out there at the beach, if the beach is open, of course. The frequency of mutation, sort of the natural rate of mutation is very low. Natural mutation rate is 1 in a billion. 1 in a billion. That's how good your DNA polymerase enzyme is. And the mutation rate will increase to 10 to the -5 or 10 to the -3 per replicated gene if there is a mutagen, a mutation causing substance or agent present. Now, there are different ways you can identify mutagens. And here are some tests, sort of briefly, we're going to talk about how you would be able to identify whether a substance is a mutagen or not. And so you can do positive or direct selection that detects mutant cells because they grow or appear different than unmutated cells. You can do negative or indirect selection, which detects mutant cells that cannot grow or perform a certain function. And then you can select for auxotrophs. These are mutants that have a nutritional requirement that's absent in the parent, and so you can use what's called replica plating for doing that. Here's how the replica plating works. You take this velvet surface right here, and you basically absorb a little bit of the bacteria that are grown on these colonies in this petri plate, and then you are stamping it down onto different plates, and then you're going to look for mutants, whether they can grow on different types of media. And here you have this mutant that is absent in this media right here.